So yesterday we had done that is what should be the genetic material based on certain criteria, and we have three contestants, that is the DNA, RNA and protein, and we have to see that is among these three which is the best genetic material. The first property is that is the self-replication. DNA can show self-replication, even RNA show self-replication, but protein cannot replicate its own. The next is stability. We know that the DNA is double-stranded. So DNA is not exposing any of its radical for the reaction with anything else. No other molecule can easily react to it. Secondly, in Frederick Griffith experiment we have seen that even he boiled, the DNA does not that is destroyed. The only thing is DNA uncoils. When it uncoils, again the nitrogen bases are there which are complementary to each other. They immediately join together as the temperature comes down. So DNA is very stable even in the high temperature, even in presence of several chemicals. Whereas RNA is not stable because RNA has only one strand, so all the nitrogen bases are exposed. They can easily react with any other molecule which are coming or any other chemical which is coming in contact with. And secondly, the thymine, the uracil is very much unstable. So uracil can easily react with something else, whereas the thymine is not. And protein, as you know, is the most unstable substance because even a slight temperature increases, the protein coagulates. Even it comes in contact with the different pH, let it high pH or low pH, it coagulates. So protein is not even stable. The third thing we wanted to know, that is the mutability. That is slight changes. RNA can show slight changes in due course of copying mechanism. Even RNA can show slight changes in the copying mechanism. Sometime or other there may be some error. Protein, since it cannot replicate its own, there is no question. Protein will always be made by either that is from DNA to RNA and RNA to protein. So mutability is also not a suitable character for protein. However, RNA and DNA both can do that. And fourth is that is the versatile nature या Mendelian characters, expression of Mendelian characters. So that is done by large number of molecules. So here again DNA can, because the DNA molecules are very long, DNA molecules are very big. So they can of course show large number of expression. RNA of course is a small molecule in compared to DNA. So they may not be as suitable as DNA. In compared to DNA they are less suitable. So I can say this is not a good criteria for which is shown by the RNA. And protein of course shows this because protein has infinite possibility. So if we consider this, we find that the DNA is the most suitable molecule which shows self-replication with the full justice, that is without showing any error. Secondly, stability. DNA is very much stable, even the physical and chemical aberrations. Mutability up to a certain extent is possible in the DNA, much less in compared to RNA. So it is not showing as occasional variation as RNA could have. And we also don't want that it should very occasionally show variation. So in that way again DNA is the better contestant, and the variable, that is the versatile molecule. So that is again is shown by the DNA, not by the RNA. However, protein can show the versatility, but then again the problem is protein is not stable. Protein is not self-replicable. Protein is not, the stability is the biggest problem. So that is why we are saying that the genetic material should be DNA, not the RNA. So 3 or 4 lectures in the previous lecture, I was only discussing on the one topic, that is what should be the genetic material. Because Mendel has said that factors are something. But what factor are, where they found, what they look like, what they are made up of, what how they express character, none of the questions he could answer. Then later on we came to know that the factors are actually located on chromosome, and as the chromosomes are impaired, the factors will also be paired, and chromosomes segregate, the factors will also segregate, and chromosome reunite, factors will also reunite. So that way factors have got some identity, that is they are there. But now still the question is that what they are made up of. So here with the experiments of Frederick Griffith and experiment of Avery McCarthy and NUCLEOID, reconfirming it, and then Hershey and Chase. Today we are in a state to say that DNA is the genetic material. Again, on the comparison of these points, we have confirmed that the DNA should be the genetic material, not the protein. And RNA was not standing only anywhere, anyway. Still we are comparing with these three. Okay. Now we must know that how a DNA or where is the DNA found in a cell. While studying about the chromosome, we came to know that the chromosomes are made up of DNA. So the DNA are found in chromosomes. And Hershey and Chase also said, Sutton and Bowry also said that the factors are located in chromosome. So again it is reconfirming this point, that is the DNA is the genetic material. And they are located in the chromosomes. Now the question is, we have studied yesterday that is there are 6.6 into 10 raised to the power 9 base pairs. And the distance between two consecutive base pairs is 0.34 nanometer into 10 to the power minus 9 of a meter. So if you multiply this, we will get the total length of the DNA, that is all the chromosomes, that is the DNA in all the chromosomes. This is suppose the first chromosome DNA, this is the second chromosome DNA, this is third chromosome DNA. Like that, if we take all the chromosomes which are found in the human being, if we then put them together, we will have, that is 48 chromosomes, then we will have the total length is equal to this. So ये कितने हो रहे हैं? 10 to the power minus 9 and 10 to the power 9, they will be cancelled, and we will have 6.6 into 0.34. So it will be equal to somewhere about 2.2 or 2.14 meter, 24 meter. So itna bada. Now the question is, the cell is microscopic. When a cell is microscopic, how such a long DNA is accommodated into a cell? Or rather, I could have put the same question in this way, that is how DNA makes a chromosome. How DNA makes a chromosome, or how such a long DNA, that is the total number of DNA which is found in any human cell, are fit in such microscopic cell. We have studied the cells are somewhere about in the micron size, whereas the length of the DNA is 2 meters. That means they have to be highly crumpled and kept. Now this crumple is the random crumpling, or it is some sort of ideal crumpling. So this we can study in the next topic, that is called packaging of DNA helix. Again here I would like to tell you one more thing, that is the DNA is negatively charged molecule, but the chromosomes are not negatively charged. Chromosomes are chargeless. That means the DNA are not, chromosomes are not made up of only DNA. There is some other molecule beside DNA which are positively charged. So overall the charge becomes neutral. So let us see. We are studying the DNA in double helical structure like this. And this double helical DNA encircle around a histone protein which is positively charged in such a way that it takes around 2 and 3/4 circle, and now it is known as nucleosome. Very important question is asked on the board. What is a nucleosome? You must know what is a nucleosome. Figure 6.4a in your book. Page number, I can't see the page. So you see 6.4a is a nucleosome. A nucleosome is having a DNA encircling around histone protein. This is histone protein. And then it is locked by another protein that is histone octamer it is, and this is H1 histone. So these two protein molecules will encircle the DNA molecule around it, and the negative charge on the DNA is neutralized. It is also noted that the histone octamer, around which the core of histone octamer, around which the DNA when encircled, it takes 200 base pairs, or it uses 200 base pairs. So this is a histone molecule, and now we shall go ahead. So I will teach you how the packing is done. The chromosome is a very complicated molecule. Let us see. This is the DNA, and then the DNA is encircled around histone, and we are seeing histone here. Then histone also makes this type of coiling. Then this type of histone coil, which is given in the book in the name of bead in the string, it further coils. And then they also coil. So how many times coiling took place? First coiling in the histone, second coiling in this core, third coiling in the solenoid. And suppose this is the centromere, and similarly it will go on like this. So this will reduce the length of the DNA to a very great extent. And this is one of the chromatid, and this side the another chromatid. So this is how it will join to it. So the chromosome will form. And in a chromosome, the length of the DNA is very much reduced. So the 2.24 meter chromosome is highly reduced in the form of chromosome. And the DNA which was negatively charged, which when circle around the histone octamer which are positively charged, they lose their charges, and this is how the chromosome becomes chargeless or neutral. So this is how the packing is done. If you see, I will just tell you the page number. It is page number 99. If you got the book around with you, you can see page number 99, where the packaging of the DNA helix is given and histone octamer is given, and then lower, just beneath that, the 6.4b electron microscope picture of beads on string. This is the beads on string. Okay, so initially when the bead on string model had come, people thought the beads are actually genes, or the beads are actually factor. But the beads are not gene, neither they are factor, but they are nucleosome. Okay, so again there is a question given to you. If the length of Escherichia coli DNA is 1.6 millimeter, can you calculate the number of base pairs in it? Can you base pair in state. 1.36 into 10 to the power minus 4 divided by 0.34 into 10 to the power minus 6. So this will go up and 10 to the power 2 divided by 0.34. So if you divide this, then you will get the number of base pairs which are found in the Escherichia coli. Okay. And nucleosome, it is asked, if the nucleosomes are asked, in the further up it is asked somewhere. The nucleosome, then you have to divide it by only 200 here. Histone of the negatively charged 200 base pair. Again the question is no the question is asked. Okay. So this is what the packing of this thing is there. And in the chromosome, you know, there are some dark phases where the genes are densely packed. They are called. So I'll read this last paragraph of page 99. Theoretically, how many such base pairs are found in are present in the mammalian cell? Mammalian cell कैसे बोलते हैं? Mammal I got so many different type of cells. What type of question they write? Instead of mammalian cell, if you consider it a human cell, you can say how many base pairs are there? 6.6 into 10 to the power 9 divided by 200. So this will divide by this. इसको इससे काट दीजिए 7 रह जाएंगे. इसको इससे divided 3.3 into 10 to the power 7 beads will be there. All nucleosomes will be there. Okay. So. Okay, now beads on string structure in the chromatid is packed in the form of chromatin fiber, and this was the chromatin fiber that just now I made. Okay, and chromatin fiber, and they are further coiled and condensed on the metaphase stage and of the cell division to form chromosomes. So we have already done about the chromatin and the chromosome in class 11 when we are doing about the cell division. The beads on the packaging of the chromatin at a higher level require additional set of protein. They are collectively referred as non-histone chromosomal protein, that is NHC, and in typical nucleus some of the region of the chromatins are loosely packed and they are called euchromatin, and some are the places where they are densely packed. They are called heterochromatin. Euchromatin is said traditionally transcriptionally active chromatin where the heterochromatins are inactive. तो heterochromatin का हमारे काम नहीं आएंगे, ठीक है. एक्सप्रेशन में काम कौन से आएंगे? ओनली euchromatins will be. ठीक है. तो with this we have come to know so many things about the DNA. ठीक है. Where तो हमारे क्या क्वेरीज पूरी हो गई. That is factors are there. ये बात सही है. Factors are located in chromosome. We came to know that factors are actually made up of DNA. That also we came to know. And how factors make, that is the DNA makes chromosomes. That also we came to know. क्रोमोसोम की दो और प्रॉपर्टीज होती है, उनको भी डिस्कस करते हैं. दिस क्लास में क्रोमोसोम हैज टू इंटरेस्टिंग प्रॉपर्टीज. One is transcription and one is translation. Reverse transcription. यहाँ देखते हैं, एक DNA खुद अपनी कॉपी बना सकता है. This is known as replication. replication. The property of making a DNA another DNA is called replication. A DNA can even make RNA. That is called transcription. And even RNA can make DNA. Only in certain viruses. And one of the famous viruses which we are encountering nowadays world over, that is coronavirus. So that is having RNA as a genetic material. First it will make RNA to DNA, and from the DNA it will make RNA, required type of RNA that will be used to make protein. So this will be known as reverse transcription. We do not have this process in our body. So the key of the vaccination of this is depending on this one. Okay, now from RNA it will make DNA, and from DNA it will make RNA, and RNA can make protein. That is called translation. Translation. चेतन भगत ने एक नॉवल लिखी, novel is DNA. Novel is printed number of times is given it to the publisher, that is called replication. Then some what you call movie maker, producer asks Chetan Bhagat, can we make a movie on your script, your book? जो भी डील उनके बीच में होनी है हो जाएगी. And then the RNA is this DNA can be changed into RNA. The RNA will become script. Novel what you are reading in all line by line, everything is there. In the script they will be divided into, एक नाम रहेगा रमेश, इसने ये बोला, सुरेश इसने ये बोला, रमेश ये बोला, सुरेश ये. So they will be in the form of dialogues. And then that dialogue can be changed into celluloid, that is called translation. Meanwhile, somebody else can see that script and they can again get it into another magazine in the different language. So that is called reverse transcription. तो ये पूरा process है, that is known as central dogma. So it suggests or it makes us understand the importance of the DNA. DNA is the key of all the expression of hereditary characters in the living being, except in certain viruses where it is not the DNA but it is RNA. So in certain viruses which are only limited characters, they can control with their RNA. Okay. But by and large, most of the organisms have genetic material in the form of DNA. Reason we already summarized. Why DNA is better than RNA? क्यों है? Because DNA has several advantages. DNA can self-replicate faithfully, कोई गलती नहीं होती DNA के replication के अंदर. वो लार्ज नंबर के कैरेक्टर्स रख सकता है, ठीक है, बिकॉज वो डबल हेलिक्स है और स्टेबल है, बिकॉज स्टेबिलिटी इज वेरी इंपॉर्टेंट थिंग. ओकेजनली इट कैन अंडरगो सम वेरिएशन और सो दैट मेक द डीएनए मोर सूटेबल. एंड डीएनए कैसे काम करता है? डीएनए पहले आरएनए बनाएगा, आरएनए विल मेक प्रोटीन एंड द प्रोटीन मे एक्ट एज एन एंजाइम and then that will help in expression. ठीक है. Now I am leaving with you another question. यहाँ पे देखते हैं डीएनए से आरएनए बनना, आरएनए से प्रोटीन बनना, प्रोटीन से कोई भी एंजाइम बनना, वो एक्सप्रेशन होना, वो सब all these are depending on one kind of molecule. They are known as enzymes. In our body, whatever physical, chemical, biological activities are going on, they all are controlled by enzymes. Okay, 1 minute. So, all our biological activities are controlled by enzymes. Now the question is, DNA से RNA बनाना है और RNA से प्रोटीन बनाना है, तो ये मुर्गी और अंडे वाली सिचुएशन हो गई. विच केम फर्स्ट? थैंक यू.